in the interior of our gene. And they need to be gotten rid of. The process of removing these introns is called splicing. So if you splice two wires together, you take two wires that are separate and you combine them and mesh them together so that they become one wire. That's basically what you do here. You're going to connect that exon to that one, and then that one to that one, and that one to that one. And so once you're finished, what you end up with is one straight strand with all four exons and no introns. You can have what we call alternative splicing, though. If you remove all three of these introns, that's the gene you get. But what if we only remove two of them and we leave this one in? We now have a different strand of mRNA that when we turn it into a protein is going to give us a different protein. So we can use one gene to make one pre-mRNA that then gets spliced differently to make multiple mature mRNAs. So we can make multiple proteins from one gene. So that So in this case, our blue in our middle is our gene. The green on the beginning and end are parts that control the turning on and off a gene that we're not worried too much about, though. This is our RNA polymerase. It's going to go along, uncoiling the DNA as it gets to it, and making RNA that kind of spits out the back end as it gets made, and then as it goes, it's going to reform the DNA strand. If I pause it there, you can see the DNA strand, the gene ahead of the polymerase, where it hasn't gotten to yet, is still coiled up. Where it's actually touching the polymerase, it's uncoiled. The mRNA is being copied off this strand and coming out the back here. Once it moves out the back of the polymerase, though, the two DNA strands come back together and is coiled right back how it started. So that's transcription. Questions about transcription? Once we've made our mRNA, we have to turn it into protein. And we said the process of turning mRNA into protein is called translation. So think of it this way. If you're transcribing something, just in real life, that word means you're basically copying what someone said, right? If you're translating something, you're taking what someone said and turning it into a different language. It's the same idea, but it's in a different language. And so transcription in us, in our case here, turns DNA into RNA. It's the same thing, it's just a copy of it. Here, we're going to though take our mRNA and translate it into something very different. We're going to go from nucleotides to amino acids. And so there are three things involved in translation. There's the mRNA, which is our instructions. We have what's called transfer RNA. It's a different type of RNA. We'll see what it does. And then there are the ribosomes, which are the organelles we talked about, which make protein. The ribosomes are going to be the thing that goes down the mRNA strand, reading the instructions, and putting the amino acids into place. The transfer RNA, or the tRNA, is what's going to bring the amino acid to the ribosome. The ribosome is going to look at the mRNA, say, I need an alanine, the tRNA is going to bring an alanine to the ribosome, and then the ribosome is going to include it into the protein. And so the transfer RNA, we say, contains an anti-codon. Remember, a codon was the base triplet, say AAG, in our mRNA. And so if this is in our mRNA, there's something that is up here. It's going to be opposite of this. What is complementary to this? What comes from an A? T, but this is RNA, and so what's it going to be? It's going to be a U. And so we have U, U, C. So this is our codon. This is our anticodon.
The word that we use here not translated into RNA anymore.
This is going to be, this is tRNA. So this is mRNA. This is tRNA. And so it's RNA, and so it has a U instead of a T. And so if this codes for one amino acid, a tRNA is going to recognize that and say AAG codes for alanine. And so there are a bunch of different tRNAs, one for each possible combination here. The one that is UUC is going to say, it's my time to go. And it's going to bring an alanine in, and the ribosome is going to glue it and stick it on the end of the protein that's being made. So this happens in 3 steps. There's initiation, the starting point, elongation, as it's making the protein longer and longer, and then termination when it stops. So in initiation, the beginning of the mRNA, the very first part of it, the leader sequence, is going to bind to part of the ribosome. A ribosome is made up of 2 parts, kind of a big blob and a little blob. And so we have the small subunit and the large subunit. Initially, they are not together. They're floating around as separate pieces. And so the small part is going to bind to the beginning of our mRNA strand. When that happens, the first tRNA, which always carries methionine, the first amino acid is always methionine, that's going to come in, and then the second part of the ribosome is going to bind. So what you're going to end up with is basically an RNA sandwich. And so you're going to have the mRNA here with part of the ribosome on top, part of the ribosome on bottom, and then right in here is going to be your tRNA carrying your methionine. It's going to put that methionine there, and then the ribosome is going to move on to the next spot. That's elongation. So initiation was the ribosome basically coming in and on either side of their RNA, locking in on it, and putting in that first methionine. That's initiation. Elongation is when this starts moving down, and it goes to one codon, to the next codon, to the next one, and the next one. So it's going to move on. It's going to read what the next codon is. The appropriate tRNA, the transfer RNA, is going to bring in the second amino acid. A peptide bond is going to form between those 2 amino acids. Remember, a peptide bond holds amino acids together in a protein, and then the ribosome moves again. It brings in the next amino acid on a tRNA, it forms a peptide bond, and then it moves. And it keeps going, and going, and going, and going, until it hits a stop codon. A stop codon does not have an amino acid that goes with it. And so essentially what's going to happen is you're going to get amino acid, attached amino acid, attached to another one, and another one, and another one, and then it's going to skip a spot. And then even if it put another one here, they're not connected anymore. And so this is your completed protein. A stop codon is a codon that does not have an amino acid assigned to it. And so it's like a space or an enter on your keyboard. When that happens, the protein that you made is going to float away. The ribosome is going to fall apart, find another mRNA to grab onto, and do it all over again. So here, we have our mRNA strand down here. This is the small part of our ribosome. This is our tRNA over here. And so this is an anticodon. We can see that AUAC is going to pair with AUG. A gets a U, U gets an A, and G gets a C. And so this part is going to guide this tRNA right into this spot. This is the rest of the tRNA, and then this blue ball up here is the amino acid. That is our methionine. So the small ribosomal subunit is going to come in. The tRNA is going to bind, and now the large ribosomal subunit is going to come in and complete the package. Now the ribosome is going to move 3 bases down the mRNA to the next codon. The next codon is CCG. So the transfer RNA that's opposite of CCG is now going to come in. And it's going to bring a different amino acid with it. It's going to bind here. And now the 2 amino acids are right next to each other. And so the ribosome can combine the 2 together and link them together. It's going to move again to the next codon, UAC. We bring in another one, link those together. And it'll keep going. And another, and another, and another, and another, until it hits a stop codon. UAA does not have an amino acid that goes with it. So the ribosome is going to keep moving, but no tRNA is going to come in, so no amino acid is going to get added, and the whole thing is going to fall apart. And so now we have our finished protein up here. It can go do its thing. The ribosome will go find another mRNA to make a protein from. The tRNAs will get reused. They'll find another amino acid to attach to, and then they'll go and make another protein. Proteins, though, a lot of times they're not finished. Some of them need to be transported. Some of them need to be put in the vesicles. Some of them need to have carbohydrates put onto them. Some need to have lipids put onto them. If a protein is headed for a lysosome, or if they're going to be secreted, they're going to be made by the ribosomes on the rough ER. Remember, the rough ER has ribosomes attached to the outside of it. So essentially what happens there is instead of the ribosome moving down the mRNA strand, it feeds the mRNA strand through it, and instead of the protein just shooting off the top into space, it actually gets shot into the ER. So as the protein is being made, it's getting synthesized and pushed into the ER. And so once it's finished, it's already in the ER. It can be packaged and moved out. So this is, I think, the final slide. This is the summary of what we just saw. So remember, DNA is a double helix, and then this is going to have base triplets, each one coding for a codon. As a whole, we have a gene. This gets transcribed into mRNA containing codons, starting with a start codon, ending with a stop codon, each codon in between coding for an amino acid. Each of these codons has a transfer RNA with the appropriate anticodon, and attached to each amino acid. Once these get put together with a ribosome, we end up with our protein. So DNA transcribed to RNA, RNA translated to protein. Questions about any of that? Any?